So far, we have briefly talked about different type of cellular junctions. Now we are going to start talking about these junctions in more detail. The first junction we are going to talk about in detail is the gap junction. So let's get there. Communicating junctions or gap junctions, as I have mentioned earlier, they electrically and chemically couple two adjacent cells. Most animal cells in tissues communicate with their neighbors via gap junctions. The gap is spanned by channel-forming proteins called connexins, and the channel they form is called the connexon. The pore size that is formed by these proteins, connexin proteins, is about 1.5 nanometers. Small molecules, ions such as sugars, amino acids, vitamins, and small cellular mediators, the signaling molecules we talked about, secondary messengers, small intracellular mediators, for example, cyclic AMP, calcium ion, they can pass through these gap junctions. But not the larger molecules like proteins, nucleic acids, larger carbohydrates, they cannot pass through these channels. There are about 15 genes in Drosophila that code for these gap junctions or the communicating junctions. Gap junctions are made up of connexins. It's a protein which transverses plasma membrane 4 times. So it is a 4-pass transmembrane protein. 6 of these proteins can assemble to form channel or a connexon, as I mentioned. They hold two cells together at a fixed distance or at a fixed gap. That's why these junctions are also referred to as gap junctions. There are 14 distinct connexins, and the permeability of individual channels can vary depending upon which connection is forming that particular channel. So here on the slide, as you can see, so there are two types of connexins shown here. One is the darker green color and the other one is the lighter green color. So they can assemble to form gap junctions in two different ways. One is homomeric gap junction or connexon, So this homomeric connexon is made up of just one type of protein, connexin protein. Whereas heteromeric connexons, they are made up of more than one type of connexin protein. Here, as you can see, this connexin is made up of light green protein and also the dark green protein. The channels they form, they can be homotypic, which means that all the proteins that are making this particular channel is the same, or they can be heterotypic. And heterotypic can also be, again, further two different types. One in which one connexon is entirely made up of one type of connexin protein and the other is made up of a different type of protein. Or heterotypic connexins can be each heteromeric on their own. Here they were homomeric. Each connexon was homomeric, whereas here the connexons are heteromeric. These proteins, the way they assemble, they form a continuous aqueous channel through which different small molecules can transverse. As I've mentioned, that these gap junctions, they hold the two plasma membranes at a similar distance, which is about 2 to 4 nanometers. And these, when they form these gap junctions, they form a continuous aqueous channel. As you can see from this cross section, this is a continuous aqueous channel through which different small molecules can transverse. The size of the pore is about 1.5 nanometers, and these junctions are formed between two adjacent plasma membranes. Each connexon is composed of 6 connexin proteins, as you can see from this diagram. Gap junctions are very important, and humans have 14 connexin genes which have overlapping distribution. Two different connexin proteins can assemble, as I have mentioned, into different type of connexons, and the properties of the channel they form vary depending upon what type of connexin proteins are forming these channels. Electric coupling, I have already briefly mentioned that synchronized contractions of hard muscle cells and smooth muscle cells of our GI tract, our intestines, is these cells which form these vessels, they communicate each other through gap junctions. And the smooth muscle contractions, which is called peristalsis, these gap junctions play a very important role in coupling these cells to forming synchronized contractions. Noradrenaline from a nerve ending stimulates hepatocytes to increase glycogen breakdown. Not all hepatocytes or the liver cells, the type of cells liver is made up of, are called hepatocytes, are innervated by the nerve. Nerve is not adjacent to all the liver cells. The gap junctions connect the cells that are in contact with the nerve with the cells which are not innervated. So the signal from the nerve is communicated to the cell which is adjacent to the nerve, and that cell communicates that signal to the cells which are adjacent to it. A specific group of cells in embryo develop distinct identities and begin to differentiate. And when they start to do that, they have to uncouple themselves. So, which basically also means the cells can regulate the gap junctions. They can also disengage when they want to uncouple with their neighbors. So they have that control to disengage from their surrounding cells. Cells in each group that remain coupled during embryogenesis follow similar developmental pathway in a very coordinated fashion. Gap junctions, as I mentioned, can open and close. Permeability of gap junctions is rapidly and reversibly reduced if cytosolic pH decreases, or if the concentration of calcium ion is very high. The significance of pH decrease is not fully understood, whereas calcium ions, we can talk about that. Generally, cells, when they're damaged, they release calcium from the calcium storage unit, which is basically endoplasmic reticulum. So if a cell has been damaged, in order to prevent the neighboring cell from the damage, the neighboring cells, they shut down their gap junctions, so the influx of calcium doesn't enter the neighboring cell. And remember, calcium is an important secondary messenger, so it will alter the function of cell if it enters the adjacent cell. So healthy neighbors of damaged cells can also change the conformation of their gap junctions, thereby shutting these channels down. So the cell is isolated, preventing the damage from spreading to the other cells. Dopamine, it is a neurotransmitter, and it's an important one. It also reduces, when it is released from the nerve endings, it reduces the gap junction communication between retina neurons in bright light. Rod receptors, remember that we talked about, they are more sensitive to light and they're good detectors. So once the concentration or the amount of light or the amount of photons increases, the cells, they switch from using rod cells to the cone cells. Cone cells can detect color and the finer detail in the brighter light. So our cells in retina, they switch from rod to cone cells when the amount of light increases in the surrounding, thereby allowing us or the animals to see color and finer detail. We are going to talk about other types of cell junctions in the later modules.